Good afternoon, everyone. Welcome to WALS. My name is Shan He. I'm a visiting fellow in NIDCD. Today, it's my great honor to introduce Speaker Dr. Mumin Pu from University of California at Berkeley, who is nominated by NIH postdoc fellows. And here, I want to encourage all the fellows to nominate your favorite scientists, and you may have a chance to host them. Dr. Mumin Pu received his bachelor degree in physics in 1970 from Tsinghua University in Taiwan, and his PhD from Johns Hopkins in 1974. Given by his physics background, Dr. Pu has very interesting perspective to study neuroscience and has been contributing enormously to different fields. In the 70s, Dr. Pu developed a number of tools to study the mobility and the localization of proteins in cell membrane. During the 80s, he developed novel uses of patch clamping recording methods in studying transmitter secretion during nerve growth and synaptogenesis. During the 90s, he made major contributions to our understanding of axon guidance and synaptic plasticity. And he made novel use of cell culture system to studying the role of neurotrophins in regulating the activity-dependent synaptic modification. His more recent studies had also been contributed to the formulation of the idea of Spike Timing-Dependent Plasticity, and this original article has been cited over thousands of times. And this is also the topic he's going to cover today. Dr. Pu is the recipient of numerous awards from around the world, just to name a few. Jefferies Neuroscience Investigator Award from NIH, Dr. Honoris Causa from École Normale Supérieure Paris, People Republic China International Science and Technology Cooperation Award. He's currently a member of Academia Sinica in Taiwan, a member of National Academy of Sciences in the States, and also a foreign member of Chinese Academy of Sciences. During his long and successful career, Dr. Pu had served several faculties in top universities at University of California at Irvine, San Diego, Yale, and Columbia, before he joined Berkeley in 2000, where he is currently Paul Leahy Distinguished Professor in Biology. In 1999, he co-founded the Institute of Neuroscience and has been serving the director ever since. This institute is a flagship neuroscience program in China and also where he is nurturing the next generation of neuroscientists. I'm very proud to say I graduated from this institute 3 years ago, so it's really great to see Dr. Pu again. And thank you for coming. Today he's going to talk about neuroplasticity from synapse to perception. Please join me and welcome Dr. Pu.
Thank you very much, San. It was a great honor to be invited by fellows. I'm going to tell you a few stories on the neuroplasticity. As you know, the most remarkable feature of the brain is the plasticity. It endows us with the ability to change in response to experience, and it makes what a human capable of remarkable ability of responding to environment and changing the environment. The plasticity of the brain starts with the activity. It turns out that electrical activity is the most potent way of changing the brain structure and function. While activity associated with sensory and motor function or associated with cognitive experience will alter the structure and function of the synapse and the neurons in such a way that the future activity through the nervous system will be altered. The learning and memory due to this experience will imprint a change in the structure of the brain so that the cognitive function and behavior will be altered. Now the question has been over the last century how the activity affects the nervous system. The idea that its direct actions on the synapse and on the neuron itself this idea has a long history, but the person who most effectively summarized and proposed the problem is the Canadian psychologist Donald Hebb. Although he himself is not a physiologist, he has been thinking throughout his career how the psychological function, the cognitive function, can be explained by physiological mechanisms. So in a book he published in 1949, 60 years ago, he proposed a hypothesis. This is known as the Hebb's postulate. In this hypothesis, he said that the perceptual experience changed the nervous system by changing the synapse. Now, he supposed that when an axon of a neuron, of a cell A, is near enough to excite a cell B and repetitively and persistently take part in firing the cell, then the synaptic connection between these two neurons will be strengthened through some changes in the metabolism and growth and structure. Now, this is what he postulated, how the activity affects the structure and function through correlated activity, activity in the presynaptic cell and postsynaptic cell. This leads to the idea of the Hebb's learning rule, where the correlated activity in the pre- and postsynaptic cells strengthen the synapse. This idea was extended in the 70s by Gunther Sten for purely symmetry, that if the activity, correlated activity strengthens synapse, uncorrelated activity might weaken the synapse. Then we have a nice asymmetry in this hypothesis. Now this idea has become very popular. We now we all say that in the nervous system, cells that fire together wire together. That this is the simple Hebb's rule. Now this is the very interesting hypothesis. It helps us to understand a lot of activity-dependent processes in the brain, especially during early development. The neural network are formed early in development, but this earlier connection will be refined by experience. And this refinement can be explained in many cases by this Hebb's postulate. Now, I'm to take you for an example, probably the best example in terms of Hebb's learning rule, is the development of the retinal-tectum mapping. The neurons that sense axon from retina, the retinal ganglion cell axon, make connection in the central nervous system in a topographically ordered manner. Temporal axon goes to the anterior end of the tectum, and the nasal axon goes to the posterior end of the tectum in a linear, orderly projection. Now, this projection is established gradually in development. In development, this projection appears to be determined by gradients of molecules in the nervous system. There's some matching of molecules on the axons, on the retinal ganglion cells, that matches the gradients of molecules in the tectum. For example, people found that the EPH receptors has a gradient in the tectum ganglion cell, while in the tectum there's a ligand, afferent ligand, which has a complementary gradient. And if the axons search out for a complementary gradient, higher receptor search for a lower ligand, lower receptor for higher ligand, to reach a steady state activation of the process that stopped the axon. One can imagine you can have a projection which is orderly arranged in the tectum. But this projection, if you look at early development, is quite diffuse. They are not very precise. And it turns out that the activity used of the visual system can shape this projection to the finer grain point-to-point projections from the ganglion cell to tectum. Now, using the Hebb's learning rule, you can understand this process. Right? For example, Hebb's rule says cells fire together, wire together. When the developing animal, in this case a tadpole, is seeing the visual stimulus from the environment, the visual stimulus tend to be correlated in space and time. Seeing an object and something flying through the space, this object in the visual space trigger correlated firing of neighboring ganglion cell because they are firing cells projected to the neighboring ganglion cell. So neighboring ganglion cell tend to fire because of correlated input together. So one can imagine that cells that fire together, these cells tend to wire together, and then their connection to the central projection will be stabilized. While ganglion cells which are far away, they tend not to be excited by an object in the visual field. They will not be firing at the same time. They are uncorrelated in activity. So the connection between these two ganglion cells to the same central target will be weakened. Right? Very, very simple idea. Correlated firing leads to refinement. So this diffuse initial projection will be refined to even a final grain point-to-point projection. Now this is a very useful way of thinking about activity-dependent refinement of the network. So 15 years ago, this is what we were faced with. We know the Hertz postulate works well in explaining a lot of things. A lot of modeling has been done. So we went the next step. We asked the question in a more precise way. We said that correlated activity of two neurons will be linked to cells that are fired together. But how correlated has these two neurons has to be in their activity? Do the activity in these two ganglion cells, neighboring ganglion cells, has to be fired within 10 milliseconds? Or have to fire within 100 milliseconds or 1 second of each other? We can define as correlated firing. So precisely how does the correlation, the requirement of correlation in terms of refinement, in terms of strengthening their common targets, the connection with the common targets. So we set out to test this. The very simple idea, we go to the tadpole, the tectum with a whole cell recording the tectum neuron. We have two electrodes placed on the retina. Remove the lens, stimulate the retina with extracellular electrode. And if you get into an early developing tadpole, each tectum cell are innervated by many ganglion cells. So choosing any two ganglion region within the retina will fire the cells effectively. The input 1 and 2 will produce very nice synaptic currents in the target cells. So they are connected monosynaptically because they respond immediately. Now if you fire these two cells together at the same time, correlated with 0 time difference, then you get a much bigger. The amplitude is the summation of these two of a synaptic current, linear summation, very nicely. So we have a situation, just what we show here, we have a situation of a cell recorded that are innervated by two distant ganglion cells by monosynaptic connection. So now we can fire the ganglion cell with extracellular recording and ask how precise have you to fire together in order to strengthen their common input to the ganglions, to the tactile cell, right? Very simple experiment. So the experiment was, now let's do excitation of two input, subthreshold excitation. Fire the ganglion cell, two regions, a number of ganglion cells are fired. The receptor potential, synaptic potential recorded in the tactile cell is depolarization. We fire these two input at a correlation of 10 milliseconds, that they are just 10 milliseconds apart, very much close in time. And you'll see two synaptic potential created. And we repeated the firing, 1, 2, 1, 2 firing for 50 times. The correlated firing within 10 milliseconds for 50 times. And then we ask, what is the connection, the response you can see in the tactile cell by input 1 and 2 after they have been correlated stimulated. The result is that there's no change. Input 1, after correlated firing, the magnitude of synaptic current amplitude remain the same. The input 2 remains the same. So subthreshold excitation that did not create spiking in the postsynaptic cell produced no effects in synaptic efficacy and under the present condition of 50 pairs of stimulation. Now, if we now crank up the stimulation, we say, well, activate more ganglion cell. We now fire an action potential in the tactile cell, still 10 milliseconds apart, now 50 pairs. Then you ask what happened to the input. The input set up both inputs. Input 1 gets potentiated, persistent potentiation. Input 2 also gets potentiated, even though it's a little bit small, but it's significant. So excitation. The firing, the Haps postulate is saying that cells fire together, wire together. Both the presynaptic cell and postsynaptic cell have to fire together. If postsynaptic cell is subthreshold, no effect. Fire together means pre and postsynaptic cell fire together. If two presynaptic cells are fired together within 10 milliseconds, both are creating pre and post correlated firing, both input are potentiated. Fits very well with what Happens to say. Now, the difference, the interesting thing comes if we now have two input, one is subthreshold. Input one subthreshold produces only depolarization, no spikes in postsynaptic cell. The second input created a spike. So now they are still 10 milliseconds apart, correlated, relatively correlated, 50 pairs. The result is that, what you guess? Well, input one, normally subthreshold input are not capable of changing the synaptic strength. Input one now gets some potentiation. Input two, it fires the postsynaptic cell. Pre and post are firing. They get very big potentiation. So Haps says this is potentiation, fits with Haps' idea. This also doesn't fit with the previous finding that subthreshold alone cannot strengthen synapse. However, if a subthreshold input is fired together later followed by a spike created by other input, not by itself, they also get strengthened. So they took the benefit of being co-activated with another input. Now the interesting thing is that if you reverse the order, you now fire the first input gives a good spike. The second input is subthreshold. The result is quite unexpected. Well, the first input, the input one gets potentiated. This is what we expect. This is a pre and post synaptic firing. They link and potentiated. The second input, which 10 milliseconds later follow the spikes, they turn out to be depressed, right? They are depressed. Both the potentiation and depression depends on NMDA receptors. It's a LTP and LTD, as you found in many other systems. So look at this situation. Two inputs are both correlated within 10 milliseconds. When the weak input is ahead of the spiking, it's potentiated. When the weak input is following the spike, it's depressed. Now the timing of the presynaptic spiking relative to the postsynaptic spiking is critical to determine whether it gets potentiation or depression. Now you can try this with 10 milliseconds pretty close. Well, you can have varying the timing between two inputs. You'll find that you get LTP when a subthreshold input is within 20 milliseconds or so before the postsynaptic spiking. If the input comes earlier, much earlier, beyond 60 milliseconds, there's no modification. If the input comes after the spiking, which is created by other inputs, you get a depression. So here, the Haps postulate doesn't work anymore. The correlation for the input is similarly correlated, both 20 milliseconds difference in two input. The order determines whether it gets potentiation or depression. So we can imagine that what happened in the synaptic refinement is that you have many input that comes to excite the postsynaptic neuron. Inputs compete with each other by firing this postsynaptic cell. The one that able to fire the postsynaptic cell, it contributes to the excitation of the cell. It gets potentiated. Now this input, even though it itself cannot fire the action potential, but it helps the second input to produce the action potential. if they are coming within 20 milliseconds, they are still contributing because synaptic potential lasts for 10 to 20, 30 milliseconds. So they made a contribution, they are rewarded with potentiation. When input comes in, when other input have already fired a cell, it comes later after the spike is already initiated. They make no contribution to excitation, they are punished with depression. Right? So competition, a synaptic refinement is a competition for timing. Now, for those input that arrive within a window of 40 milliseconds, roughly, right? The one that comes early among these input that arrive within 40 milliseconds, those come early gets rewarded, those come slightly later gets punished by LTP and LTD. Those comes too early or too late are out of the game. No more, they are not even affected. Right? Competition is a competition for timing. So this idea leads to the idea that this finding leads to the idea that temporal specific Haps rule is a better learning rule, where the correlated activity not only can strengthen the synapse but also weaken the synapse, depending on the temporal order of spiking. If the presynaptic spiking comes before postsynaptic spiking, it's strengthening. The reverse order is weakening. So we call this spike timing-dependent plasticity. Now, when we found this, actually, this finding, this window, Henry Markram and Bert Sackman already had a report in the cortical pyramidal cells, where they show if they fire the postsynaptic neuron with the action potential that backpropagate into the dendrite, that arrival of the backpropagating dendritic potential, action potential, if it comes after the synaptic input, it will potentiate the input. If it comes before the synaptic input, it will depress the input. So they had this experiment with a 5 millisecond or so before and after showing potentiation depression. So we said, well, what we are seeing here, instead of firing the postsynaptic cell, we actually synaptic input-driven spiking fits the same rules. In fact, we also tested by current injection to fire a spike in the postsynaptic cell with a timing relative to the input. And using current injection to trigger the spike, it gives the same window. So this appeared to be a general phenomenon, not only in the early developing tadpole brain, but in many other places. Now, over the last 10 years or so, there are many systems where people tested this in vitro, in site culture, even in vivo preparation. It turns out that the LTP is always created when presynaptic cell fire before postsynaptic cell, with a relatively narrow window of 20 to 40 milliseconds. While the LTD is often created when the postsynaptic cell fire before the presynaptic cell. Right? So the negative time interval, the window appear to be much more variable depending on the synaptic type, can vary from 20 up to 100 milliseconds in different synapses. So this appears to be a relative general rules. Now, this spike timing-dependent LTP and LTD uses mechanism very similar but not identical to the previously known LTP and LTD in the hippocampus. I wouldn't go into detail, but they are also depend on NMDA receptors, but the mechanism and expression of the LTP and LTD are slightly different in different synapses. So what does this mean? Why would it be useful to have a synapse that detect the timing of the pre and postsynaptic cell? I want to play a game with you to strengthen your appreciation how important the timing is. Right? I'm going to play two pieces of piano music, and I ask you to tell me what tune it is. Right? The first, a classical composer. Some of you might recognize it, but I guess most of you will not. That's my experience when showing this. But I want to play the second tune. I'm sure most of you recognize it. This is the piano piece by Beethoven, right, for Elise. Now, it turns out that these two pieces are the exactly same piece. The computer played the scores backwards in the first tune. You totally cannot recognize it. The interval exactly the same. It's the order of the key is reversed, right? So this tells you that the memory, our memory, is highly temporally specific. Right? It's specific for the sequence of events. Each tune, each note comes in with a sequence, and also the interval between the notes, the time interval of events. So this is a typical memory. Episodic memory works this way. You always remember what you do first, and the second, the sequence is always there. The time interval is also there. So how does the nervous system code this? So I'm going to show you a few experiments that tells you that one can use this spike timing-dependent plasticity to code the temporal sequence and interval of the information for memory, for learning. So the first experiment uses the mouse visual cortex. Now I'm going to show us a simulation of a simple model that's supposed we have in the cortex, groups of neurons are connected with random connection. There's no bias. Let's say this is one group, second group, third group, they're all connected to each other in recurrent connection, forward and backwards. The strengths are all the same. It is a starting condition. Now, if I give a stimulus that gives you a sequential excitation of this group of this ensemble of neurons in a sequence from left to right, so the result would be that the synaptic connection would be modified. Right? So we will fire the pre-post. For the connection, the forward direction connection, the synapses are seeing pre-synaptic activity before post-synaptic activity. So they all become strengthened. While the reverse synapse all become weakened, according to STDP, right? We all agree with that, given the proper timing of excitation of this group. So we have an asymmetric, after training of a unidirectional sequential activation of in this ensemble of neurons, we create an asymmetric connectivity based on STDP. So then, if we now give a partial stimulus, let's just excite, only give a partial stimulus at the initial point. Because of strengthening of this group, you tend to fire the next group of neurons. So a partial stimulus can now evoke a sequential firing of the group that are connected by during the training. Right? So one could imagine that the memory are stored in this connectivity. It can be recalled by partial stimulus. So can we show that the cortical neuron might be using intracortical connection to carry out this function? Right? So the experiment was done by two former graduate students, Senjin Xu Senjin and Jiang Wancheng, using red V1 and a multi-electro recording on the V1, which are mapping the receptive fields of this this primary visual cortex. Now, so here are the maps that for 16 electrodes, that they are responding to different regions on the visual field, and the map region you can approximate by these circles. And if you record from all these neurons, these 16 electrodes, you'll find that according to their receptive field position, they are fired more or less in a sequence. Right? Those closer to this starting point of stimulus tend to fire first, and then they are firing in a sequence. We can actually depict this sequential firing by coding the number of spikes by the gray levels of these circles. So that this is what we will get during conditioning. A visual stimulus, a moving spot is moving across the field, And these are the receptive field of the electrodes, the 16 electrodes. So what we are seeing when we sweeping during the conditioning, we're conditioning the firing in a sequence among these 16 group of neurons being recorded. So this condition presumably will create a memory. This memory would be inherent in the connectivities within the cortex. Then we can recall this memory, recall the sequential firing by firing only the starting point, only a flash, rather than a moving spot. Right? And the flash on the end point should not create a sequential firing as effectively as the starting point, because the connectivity are biased into one direction. Right? So this is what we are expecting. So this is one example of the result. So we starting with the experiment by testing a flash on a presumed starting point of a training point. This is the rosters of 16 electrodes. Each black dot representing one action potential. So there are a lot of spontaneous activity going on. If we flash at a starting point of this group of ensemble of neurons, you'll see there's a lot of activity which appear to be random. Now, if we then give you sequential activation with a moving spot, then this activity start to be more sequential. The one that activated earlier gets fired earlier, then you get a sequential firing of the set of neurons, right? So this is the training. So after training, we go back to test with the flashing only at a spot, see if we can recall this activity, and the answer is yes. In the 7 trials, out of 7 trials, in this case, at least 4 out of the 7 show very nice, maybe 5 out of 7 show very nice sequential spiking. That was tested after, within a few minutes after we have trained, conditioned the cortex. So we can do a lot of analysis, which I will not bother you about, looking at correlation of the spiking to show the sequential spiking. And the analysis using this sort of cross-correllogram, showing that if after the conditioning, we have this bias of activity toward, or there's a slant in activity in the correllogram, showing that there's a sequential firing. The slant to the right showing sequential firing following the same sequence as the conditioning spiking, and either in awake animal or in the anesthetized animal. So this increased spiking, sequential spiking, can be recalled by flashing in the beginning points in both awakened and anesthetized animals, or in the middle of the conditioning path, but not in the end. If you flash at the end, you don't see increased correlation. If you flash with a whole bar rather than a sequential activation, so a bar conditioning produce no sequential activation, then after that you don't see this increased correlation, increased sequential firing. So all this suggests that indeed there's a memory of sequential firing created by a unidirectional sequential activation of the cortical neurons. And this firing, lasts for a couple of minutes. It's not very long. In fact, in awake animals, by 6 minutes after conditioning, it appeared to be not significant. So a few minutes of memory of a sequential information, presumably by spike timing-dependent plasticity. Now, the question is the spike timing of the cortical neuron sufficient and necessary for the cortex to learn this sequential information? So we can now test with the new tools of optogenetics tools, test whether firing the neuron directly rather than visual stimulus, firing the V1 neuron directly in a sequential manner, can you imprint a sequential firing into the circuit? Right? These 2 graduate students, Yan and Zhang, are doing these experiments using viral transfection of a channel rhodopsin into cortex And then putting electrode, you can record along this cortex that express in channel rhodopsin using light activation to activate neurons directly in a sequence. And right after the conditioning, you can test whether you can recall this sequential firing. Now, with the optogenetic stimulation, you can see this firing, the spike roster, or this PSTH, the number of spikes triggered by the firing is much more regular than visual stimulus because we're really controlling the group of neurons. You can see a nice sequential firing of the cells recorded by these 8 electrodes. Right? The answer for this is before the conditioning, testing at the end point or beginning point, you do not see any sequential firing. But with conditioning, as I showed you before, then you test again. You begin to see this sequential firing. So this is an example. Before the conditioning, you look at the group of electrodes that are recording different neuronal ensemble. This is a multi-unit recording. There seems to be random, no sequence. Then conditioning imposes a sequence, right? Conditioned by light, right? Channel rhodopsin activation give you a sequence. Then after conditioning, you test with a flash in the beginning. You start to see some clear sequential firing in many cases. So activation of neuron itself is sufficient to imprint a sequential information that can be recalled by partial stimulus at the starting point of this sequence. The clear example of increased sequential firing after conditioning for the starting point flashes, but not for endpoints flash. That shows no increase in sequential firing. The average for many cells shows the same result. So there's still a question to be addressed. Is the firing can create imprint a sequential information? Is the spiking itself required? Whether it's necessary for visually induced memory? Right? You have a visually induced, moving spot induced sequential memory in the brain. That really depends on the cortical neuron firing, right? So one can do that experiment now with channel rhodopsin experiment to inhibit the cortical neuronal firing while training with a visual stimulus. There are other questions. We are assuming that this is due to spike timing-dependent plasticity, LTP and LTD. We have to show that this visually created sequential firing can really promote, potentiate LTP of intracortical connections in a unidirectional manner, and LTD in opposite manner. Right? This is meant to be shown. There are brain state. We have shown experiment which I didn't tell you about. That brain state, whether the cell is the cortical in a awake animal is in a awake alert state or in a drowsy sleepy state. Their ability to recall the memory, the sequence memory is different. Right? The sleepy state appears to be easier to recall the memory. Well, whether the learning of the sequential information also depends on states is not clear, so being tested right now. We also show this sequential activity as a linear activation of a group of cortical neuron. Now, if we have a nonlinear activation, random activation, but in a fixed sequence of a random ensemble, would those sequence firing still be recalled among this randomly selected nonlinear group of ensemble? This is actually a very important experiment to do. While finally, we are talking about a short-term memory of a few minutes. Whereas we remember sequence information, the example I show you of the piece of music, the sequence of notes. Those are somewhere in our brain somewhere. We don't know where they are. Are they in the cortex or is somewhere else? Is the sequence information created in the cortex a temporal storage, or do they later get transferred to other places? Those are the unanswered questions. So I think I'm rather slow in presenting my story, but I will tell you another story about how the interval can be coded. Well, I talk to you about the sequence. Now, in this study, we used a randomly connected hippocampal neuron in culture, rather than a random connection assumed in vivo. Now, this you can have a group of hippocampal neuron culture in the dish. They are all randomly connected. Everyone is connected to everybody else, basically. So in this case, you can actually create plasticity, create a LTP or LTD by firing the cell sequentially. You can fire this before that, then the connection from this cell to that cell will be strengthened. LTP and LTD are demonstrated very nicely asymmetric spike timing-dependent plasticity in the culture system. So now we ask, can we use this spike timing to store the memory of time interval? Now, what do we mean by time interval? Well, here is an example how this culture could work. Here is a culture with a randomly connected connection mediated through many other cells. You have a cell being stimulated, a cell recorded in the culture. Now, because they go through many pathways to reach the other cell, if you excite one cell, the response you receive in the recorded cell could be polysynaptic, that goes through many synapses. So each of these peaks, this is actually inverted synaptic current. The synaptic currents of each peak represent a transmission of the action potential signal from one pathway that produce a delay of the first one about 20 milliseconds to reach the second cell. For the second pathway, it would take longer to reach the recorded cell. The third pathway, even longer. So by looking at polysynaptic, reproducible polysynaptic response in the neuron, you actually are monitoring pathways, specific pathways in this randomly connected network. So these are actually pathways are very stable. For an hour period, we can present this amplitude by the color. The whiter color or red colors means higher amplitude with time. Right? This is one trace here will be one trace here. Right? So your first peak, second peak, third peak. And with time, these peaks are stable. So it's saying that in the culture, it's actually quite stable connectivity, the patterns connected. So now we ask, what happens if we give pattern information into this with a certain interval information into this system? Now, can they repeatedly leave imprint in this randomly connected cell? What is the simplest interval information? Will be a pair pulse, two stimulus with a fixed interval, right? So this is an interpulse interval of a fixed duration, fixed interval, and repeat that again and again, right? So that's the simplest interval information we can imagine, right? So now let's fit in this simple interval information into the network and ask what happened to this information. How are there's mechanism in this network that remember this. Now we can do a thought experiment, a Duncan experiment first. Let's think what could happen if you give pair pulse to a cell. Now here is the cell that we stimulated with pair pulse. Suppose this cell had a two pathway to link to another cell that's recorded. The first pathway is shorter. The second pathway is longer. Now, when we give the first stimulus of the pair, the first stimulus, that's a red stimulus, gives you two signals in the recorded cell. One is a subthreshold depolarization. The other, after a delay, longer delay, you get a spike. So the first pulse gives you these two red traces. The second pulse, after the interval, you give another pair, a subthreshold depolarization and a spike, identical, right? So because of this interval between this pair pulse, you will determine how these signals received in the target cell is going to interact. If you choose the interval right, then the subthreshold stimulus produced by the second pulse fall before the spike created by the first pulse. Then you get a potentiation of synapse at that site, LTP. Right? If you change the interval, make a longer interval, you might have a situation where the input falls behind the spike. You get a situation of LTD. Right? So by different interval, we'll be able to create LTP or LTD at a very different site along this network. Somewhere will be LTP, somewhere will be LTD, depending on the interval of the pulse. Now, can we find that? Can we see that difference? How do we see that? We can look at the polysynaptic response. So here's the simplest one, simplest experiment, where we have only, we can actually have one electrode in one cell, stimulate an electrode, and record a response after a feedback to that cell. Right? So we eliminate the recording electrode. This is the simplest case. So we have a recording artifact, followed by, after stimulation, you get a synaptic currents recorded in the same cell. This comes after a circuit that activates the cell. Right? Then after this is a stable recording. We give repetitive pair pulse into the system. And after 50 pairs, actually 20 pairs of a 50 millisecond interval, what happens is there's new peaks appeared when we again stimulate the same cell. What does this new peaks mean? It means a new pathway being activated with a delay of a longer delay, two different peaks. These two peaks have a longer delay, represent new pathways which are longer that are activated. Now we have another example where we have a polysynaptic peaks after repetitive stimulation of 30 millisecond interpulse interval pairs, right, 2 pairs, for 20 pairs. After this interval stimulation, this early pair, some of the peaks disappeared, and the pattern changes, right, suggesting the polysynaptic pathway have been modified. Depending on different intervals, you have a different modification. Sometimes you have increased reliability of the polysynaptic response. Sometimes a new response appeared. Sometimes response disappeared. Polysynaptic response disappeared, suggesting pathway are turning on and off by this different interval of stimulation. Right? So this is what, if you use a cologram that we represented, you will see the same cells. We are using one single cell, use both stimulation and recording. There are many pathways that feed back to the same cell. In the beginning, with a 60 millisecond interval, pairs, 20 pairs, produce no permanent effects. We have 3 peaks that seem to be stable, 2 peaks that seem to be stable. Now we change to 60, same cell. We now give another 20 pairs of 40 milliseconds instead of 60 milliseconds. Immediately, new pathways are turned on that are stable for an hour. Right? We now create a new pathway within the system. Right? So this is consistent with distributed LTP and LTD. Right? They're consistent, but to demonstrate whether in particular site there's LTP and LTD, we actually performed a really tedious experiment to show that's actually indeed happening. Right? So this is reproducible. It's not as random, because here you see that 100 milliseconds produce no effects, 50 milliseconds produce a great change, and 20 milliseconds produce further change. So in short, the answer from this, the conclusion from this set of studies is that memory of time interval up to 200 milliseconds can be stored in a network of 20 neurons, culture neurons, By the way we assayed it is to look at the changes in the probability of these polysynaptic peaks that are modified after pair pulse of that particular interval for a number of pairs. Many of these changes, most of them are dependent upon AMDA receptors. If you block with AA, you use APV block, very little change will be observed. So the idea is that the temporal information can be specifically distributed as LTP and LTD by spike timing-dependent plasticity at synaptic site, various synaptic site within the network. Right? Exactly where depends on path length. Right? So is this a good model? Is this a realistic model? In real life, in vivo, you excite one neuron, you don't fire the next neuron because the synaptic input from one neuron to another neuron is always subthreshold. But you can fire a group of neurons. You can always fire the next group of neurons. It's called a syn-fire chain. Right? So in this culture model, we are actually saying that each culture neuron, because of their enhanced connectivity, they actually represent a group of synchronous firing neurons. What we're showing is a syn-fire chain of groups of neurons activating each other through different path lengths. And that path length can be used to store information based on the delay, synaptic delay. Right? In this case, the longest we can see changes about 200 millisecond interval can cause changes in this polysynaptic current. So at least the intervals of this short time actually can be memorized in a circuit. So how about longer time interval? What would that happen? Now in the original HAPS postulate, HAPS says the way that perceptual information is ingrained in the brain is actually to create a activity among a group of neurons that he called a cell assemble. There's activity created by the perceptual experience that would last for a while. They called reverberating activity. And that reverberating activity is the activity that fire the cells together, that keep the long-term imprint. Right? So perceptual information might create some persistent activity in the brain that are lasting longer than the presence of the perceptual experience. Another reverberating activity would be the mechanism to strengthen the connectivity among this set of cells. Now this is a HAPS postulate. Now over the 60 years period since HAP, people have looked for the reverberation in the brain. Most people said, no, there's no reverberation. It's hard to find it. Right? Perceptual, you could not see reverberating activity still going on in the circuits after the experience is gone. Right? So we decided a few years ago to test this. Herman Sombrit actually test this in zebrafish with a large group of cells in the tectum with calcium imaging. Here you can see many cells, whether there's activity, residual activity after experience that can be monitored by calcium activity. The calcium activity reflects the spiking of the neuron. Each calcium transient reflects the number of spikes. Right? They are directly proportional to the number of spikes. It's an integrated response of spiking. So if you take a tectum, you give a stimulus to the zebrafish by, say, a sweeping bar across the retina. You will see many neurons are active. This is 4, 5 neurons. Some neurons are not active, but many neurons, every sweep of the moving bar across produce a transient. They are going very nicely, following the sweeping of the visual stimulus across the retina. So what Herman was looking is, would this visual perception of repetitive bars across the retina create activity in the ensemble of cells that are lasting longer than the stimulus? Well, he found some. Here's some example. Well, firstly, he showed that this activity in the tectum are highly specific. This is a representative of calcium imaging. Calcium activity reflects on 180 cells are very reproducible. For leftward moving bar, they create a set of firing that's different from a rightward moving bar. Some overlap, but a lot of are specific to that direction of the bar. They are reproducible. You go back to the left forward bar, they actually reproduce the same firing, right? So a different ensemble of cells are responding to leftward perception, rightward perception. Some are used for both left and right. Okay, that's very simple, right? So the question is, what happened? So what he found is that after the perception, after the leftward 20 times of moving conditioning, he stopped the conditioning and watched the calcium activity. He found that there was a repeated correlated calcium activity after the bar, after conditioned stimulus has stopped. Here's again repetitive activity. Not too many cases, but it would be a few repeats after the conditioning. The interesting thing is that this repetition of the repeat is exactly the right interval as the conditioning response. So the conditioning of 4 seconds interval. After termination of conditioning, 4 seconds, 8 seconds, you get activity. You use the 6 second intervals. After conditioning, you wait for 6 seconds, 12 seconds, 18 seconds, you get a calcium activity. Calcium activity reappear with the right interval as the conditioning interval. And the 10 milliseconds, you have a repeat. After 10, 20, in one case 30 milliseconds, you get an activity. Now this is very striking because you think about the fish actually are remembering the beats of the stimulus that are once every 10 seconds you get a stimulus. Once 10 seconds you get a stimulus. And the fish actually remember 10 seconds later a beat will come, and by showing the calcium activity. So time is almost up. I'm going to show you a fun movie showing how this affects the behavior of the fish. The behavior can be the fish larva can be immobilized on a microscope stage. We can look at their tail, allow their tail to move, so their tail can swing. The head is fixed. We can monitor the calcium. So we ask when the calcium activity has oscillations after the conditioning, what happened to the fish behavior? Well, during the conditioning, every flash, in this case is a flash, every flash produces a tail sweep. The tail sweep is a response of the tail. You can monitor the tail movement, right? So the tail, 60-70% of cases, every visual stimulus create a tail response, an escape response. But after the conditioning, there's wait for two more interval, 6 seconds and 12 seconds. There are 2 more sweep, exactly at the time of 6 second interval. The tail sweep again. Right? This is exactly the time, if you look at a calcium imaging, the wave of calcium reappear on the same group of cells that's being conditioned. Right? So let's show you, this is really how good the tadpole is remembering. I only show you the last 4 conditioned stimulus. Every 6 seconds. The sound is, I put in the sound. The flash is the light. That's the stimulus. Sound is for you to recognize the time. You see that this is the last conditioned stimulus, and there are no more stimulus. Wait for 6 seconds. No stimulus, 6 seconds. I bet the fish is timing better than you are in keeping the time. And this time is exactly the time where the calcium oscillation goes up. That's that group of cells. So I think I have 2 minutes left for my time. Is that 2 minutes? Well, I really want to show you a set of experiments. This is really the high-level thing. when we talk about the perception. The perception, the most high-level perception is self-awareness, consciousness. We know we see a mirror, my face in the mirror. That's me. I am aware of myself. Now, only humans, a few species of great apes, are known to recognize themselves in the mirror. Human baby takes 2 years in order for them, in general, to acquire this ability to see themselves in the mirror. Right? So this may be acquired ability. Some psychiatric patients and autistic kids lost this ability of recognizing, lost of self-awareness, doesn't pay attention to the mirror, probably doesn't recognize themselves in the mirror. So the question is, where is the origin of this self-awareness? Or the more specific, mirror self-recognition is acquired, can it be trained? If there's impairment, can we reintroduce, we can repair cure the impairment by training, right? This is the monkey. Rhesus monkey cannot do this. They cannot recognize them in the mirror, right? So, well, the human brain, as we know, between the 2 years of age, there is this tremendous growth of network. During this growth, many abilities are appearing, right? Is it possible that the acquisition of the self-awareness is because of this time through learning? So we recently, this is totally a preliminary, non-published, I think the Gonglen and Zhang Li Tan agree for me to talk about this, even though it's unpublished. Right? This is a fun experiment, you see. What we decided to do is to train monkey to learn association of an image in the mirror. This is an image in the mirror. The monkey is sitting on a chair. We learn association of the visual and somatosensory association first by shining a laser light at a higher power that gives some feeling, some probably tinkling feeling. Or if you put on a human, I feel a little bit of heat on the monkey. So when monkeys see this image in the mirror, he feels a sensation in his own face. Now this can be very easily trained. The monkey, very quickly, they would touch that point, right? Now we give the food in order to encourage them to touch the spot, because afterwards we are not using the high power laser. After a few weeks of training, you now use a very low power laser. It produces no feeling in humans. They would do this. They would look at the mirror and then they touch the corresponding spot. Now, because they have been trained to do this, they can receive food on that. So that's why the food reward is used for in the beginning. Now, but the real standard test in the field since 40 years ago, Gullop have demonstrated in the apes can have self-recognition in mirror, but not monkey. 40 years ago, the standard test is to mark test, is to mark on the face of the monkey a dye and see whether monkeys look at a mirror and then touch that dye. This is called a mark test. The mark test is a mark on the monkey's face, and the monkey would to touch after the training. Now, no food. Now they are not rewarded. They just look at the face, different mark, different color. There's green dye on the face. They would scratch and look, right? And this is the monkeys. 3 monkeys all learn very well. Before, without mirror, they never touch. With mirror, they touch the spots or touch the dye mark. Now, this is not good enough. You have to show the spontaneous activity, not on the chair. They're spontaneously whether they look for the mirror and doing mirror-directed behavior, right? So, so this is first we do dye task in the well-trained monkey in a home cage with a mirror. They appear to look. We actually find 3 monkey trained, only 2 actually can do this in the cage. 1 monkey failed to be interested in mirror in the cage, in the home cage. And the time they look, the time they touch the face is always the time they look at the mirror It's not a coincidence, right? They can use a spot. They can follow a spot. This spot also produce no sensation. Different colors, they also follow the spots. They very nice response in all three monkeys, right? So now, the really final critical test. Are they looking at a mirror, they touch the face, it's a conditioned response. Any face with a monkey face, with a dot on the face, they'll touch because they were trained to do this, right? Are they really seeing that as themselves? Well, one way is that you put another monkey in there with a face mark on the face, and the monkey look at each other. They look at the other monkey with a face mark. Do they touch the other monkey or touch themselves? That's one test, right? So cohorts with a naive monkey with a face mark, right? This is one experiment. So cohorts with a naive monkey, which is a yellow color. So, the naive monkey has a mark on the face, and these trained monkeys doesn't touch his own face. He touched the face of the naive monkey. Right. And then when they look at it, when the mark is on its own, it would touch on its own. The trained monkey would always touch its own face. Right. Now finally, the test is a better test. Now, before I say that, so we have another monkey. This we call the glass mirror test. We alternate a transparent glass and a mirror with two monkeys, both mark at the same position, corresponding position. So the trained monkey is looking through a glass, another monkey with the same mark. What would happen? Does he touch his own face or have social behavior? The answer is they have social interaction. They never touch his own face. But then you interleave with a mirror. Half the time with a glass, half the time with a mirror. We repeat it back and forth. When the mirror was presented, the monkey trained monkey would touch its own face. Right? This is we call the mirror glass test. With a glass wall, the only time they appear to is quickly, so they have social behavior through the glass without touching. Now the mirror is in. So this worked. That the glass mirror, it seems to recognize themselves. So finally, not on the face, but in the other area, they have never been trained, like ear. They would look at this trained monkey would look at regions that have not been. They would look at themselves at the places where they are curious about. The naive monkey. Okay, implications. Rhesus monkey can learn mirror self-recognition. Human babies may acquire the ability via learning during the first 2 years. Impairment of self-awareness may be treated with training. Okay, these are people who contribute to the work. I mentioned most of their names, both in UCSD and Berkeley, and now in Shanghai. Thank you very much.